look at just some of the results of these animal-human infections. So viruses that cause lysis of their host cell, just like we have lytic bacteriophage, we can have lytic viruses, or they're sometimes called cytocidal viruses. So folks, what does the suffix cytal mean? To kill, yeah? And cyto means cell, yeah? So these lytic animal viruses, we could call them cytocidal viruses because the end result is what? Death of the host cell? Yeah, okay. So, we can think of this as occurring in a couple of ways. Sometimes the viruses, remember how they redirect the cell's biosynthetic machinery just to make viral proteins and viral nucleic acids. Sometimes that will lead to an early cell death because the cell can no longer repair itself. Yeah? So that could cause an early death and lysis. Or certainly in viral infections where they cause so much damage to the cytoplasmic membrane that the cytoplasmic membrane, the lysis breaks open. And folks, this is just, obviously I was trying to get the most dramatic example, but this is an adenovirus that's causing a lytic infection. So you see incredible inflammation of the eye, and this looks like it's actually hemorrhage, right? Hemorrhage from the blood vessels. This one you guys we're not going to worry about right now. Just forget this one for right now. Because this one is a far more important topic. So, humans have been co-evolving for viruses, since we've evolved. And so there's definitely been natural selection for humans that have the ability to protect themselves against especially lethal viral infections. In our specific acquired immune responses, a powerful strategy for protection is to produce antibodies or immunoglobulins that can bind to specific pathogens, to bind to their toxins. So we want to see how production of antibodies can protect us against infection with viruses. So we'll use this, folks, as our generic human cell. And these little cups up here, these represent the specific host cell surface receptors to which the viral adhesins will bind. And we'll focus first on the virus. Here's a generic virus, and we can see these little lollipops sticking out. The lollipops, the tips represent the viral adhesins. And so for this virus to attach to our cell, to absorb, the viral adhesins have to bind to these complementary host cell surface receptors. These little Y-shaped molecules are our classic cartoon for antibodies or immunoglobulins, right? So if we have made antibodies that can bind to the viral adhesins, as we're seeing here, would that block attachment?
Yes.
Yeah. It would, right? You're physically putting a barrier between the adhesins and the host cell surface receptors. So as a result, folks, those antibodies would block attachment of the virus. Can the virus infect our cells if it can't attach? No. It's so beautiful. It's so elegant. So antibodies that block attachment, we give them a special name. We call them neutralizing antibodies. And it's just like one of the most beautiful, efficient ways to prevent infections by bacteria, to prevent infection by viruses, by bacteria. It's also cool, folks, because when we start talking about bacterial protein toxins, exotoxins, we can also make antibodies against bacterial toxins. So by binding to the toxin can't bind to our cells, and thus no harm will result. So important vocabulary, folks. If I ask you, what are neutralizing antibodies? These are antibodies, that's right. These are antibodies that block attachment. And again, folks, we just want to remember they can block attachment of viruses, they can block attachment of bacterial pathogens, they can block attachment of protein toxins. And we'll see in chapter 14, 15, such toxins we call exotoxins. Okay. So many of our vaccines, one of the goals of the vaccines is to trigger production of neutralizing antibodies against a pathogen or against a toxin. Right? So in chapter 14, 15, folks, we'll talk more about vaccines and antibody production. we'll give some specific examples, of neutralizing antibodies triggered by vaccines and how they can protect us. Yeah, so this is just so cool. And then another really important topic, folks, is, why is it so hard to treat viral infections? With bacteria, at least we have this arsenal of antibiotics. Why is it that we have so few magic bullets for viruses? And I think there's at least two reasons. And one reason is we want to remember the virus is using our own biosynthetic machinery to replicate themselves. So the problem is, how do you stop viral replication without also stopping the important biosynthetic machinery of your cells, right? So it's hard finding, these special targets, unique viral targets for viral magic bullets that will only stop viral replication without also causing harm to our cells. And it's true, folks, some of the antiviral drugs, they have really serious side effects, really unpleasant, sometimes really serious, because by inhibiting viral replication, we also end up inhibiting normal cell function, right? And then another thing, you guys, I think is maybe another reason, is if you think, where do the viruses replicate? Where are the viruses replicating? Inside, right? So here are the viruses. The viruses are replicating inside the cell. And if you think about most of our drugs, most of our antimicrobial drugs, they're water-soluble, hydrophilic, right? So we would have really high concentrations of our antiviral drugs, say out here. In the extracellular compartment, we might have pretty high concentration out here. But this is a common problem, folks. Very often, the cytoplasmic membrane is a diffusion barrier against our antimicrobial drugs, right? So very often, it's really hard to get, because the cell membrane acts as a diffusion barrier. It's often really challenging to get therapeutic levels of your drugs inside the human cell. So I would argue that the viruses are also protected by replicating inside the human cell. It is a bit of a protected little ecological niche for them. Yeah. So just in general, folks, one approach has been for antiviral drugs. Some of the antiviral drugs that maybe you've heard about, for example, ACT was the first antiviral drug used to treat HIV. And another one we'll be talking about herpes viruses, acyclovir. A strategy is to use what are called nucleotide or nucleoside or base analogs to try to selectively slow down viral genetic information replication. And these analogs are look-alike. They look like the normal nucleotide, nucleoside, or base, so that the RNA polymerases or DNA polymerases will incorporate them into the viral genetic information. But they are abnormal in some way. So for example, some of the analogs will lack a 3' hydroxyl group. And I know you guys, you're probably like, what is she talking about? But if you go back and look at DNA replication, we said you have to have that free 3' hydroxyl group for the incoming nucleotide to be added, right? So one approach is that you use an analog where there isn't a hydroxyl group on 3', it's just a hydrogen, right? So when it gets incorporated into the end of a growing strand of nucleic acid, it stops the synthesis there because you can't add any more nucleotides. So they call that premature chain termination. And those special analogs are called dideoxy for two oxygen missing dideoxy nucleotides. So those can be used to cause, they call it premature for early chain termination. And then others of the analogs, they may be incorporated, and then when that strand is used as a template, that analog might cause incorrect base pairing, so it introduces lots and lots and lots of mutations. And the idea is you're hoping you can disrupt the structural genes for the viral proteins, right? So the proteins won't work. So that is a one approach, one strategy for developing antiviral drugs. But the problem is, you guys, is that some of these analogs, our own, polymerases will use them, and so that's why often there are these really serious side effects. Is our polymerases will use them too, and that we can end up with adverse side effects. Yeah. And this is just introducing a couple of concepts, some special features of some viruses that we'll elaborate in the next PowerPoint. But let me give you the background story on this, you guys. So a few years ago, we were so lucky. We had one of the emeritus microbiology professors from the UCD Med School came to give some guest lectures. And his name was Dr. Robert Chang. He was born in China, educated in Shanghai, had this beautiful, elegant British accent, and dressed very formally, suit and tie, just, really, really lovely person. And so he was giving the lecture on sexually transmitted infections, and so we were approaching the herpes viruses, some of which we will see can be transmitted sexually. And so he asked the class with a straight face, he goes, "Now class, why are herpes like diamonds?" And I'm the instructor, right? I'm like, I'm supposed to know this stuff. And I'm racking my brain. Why are herpes like diamonds? Why are herpes like diamonds? And he says, because diamonds are forever. He delivered it so straight face. British accent. I go, I can't believe he said that, right? So, but it is helpful, folks, because we're going to discover that there's many different types of herpes viruses. Some of them are like diamonds, because once we're infected for the rest of our life, right? So that's the idea there. And why am I bringing this up? This concept of a virus causing lifelong infection, and part of this, folks, it's not just a lifelong infection, but it's that we're infected and we don't even know it. We don't even know we're still infected, right? We refer to these lifelong infections as latency. And some of the viruses we're going to be talking about are notorious for causing latent infections, these lifelong infections. And what I'll do, folks, without going into a lot of this, for example, the herpes viruses, what the herpes viruses will do, they'll first replicate on epithelial cells of the skin or mucous membranes, and will have, very often painful, really distressing lesions. But then eventually the immune response responds, and hopefully in a few weeks, most of those painful lesions are gone. And we're like, whew, wow, I'm glad I got through that one. But what we don't know is, with some of these viruses, is what they've done is they've invaded our sensory neurons. And they're actually residing in the neuron cell bodies. They're not actively replicating. So we don't have any clinical signs or symptoms. We think we're cured, right? But then later on there can be a trigger. Usually it's some kind of stressful trigger, something that suppresses the immune system. It will trigger, reactivate the viruses, and what they'll do is they'll travel back down the sensory neuron and they'll invade the epithelial cells that the sensory neuron innervates. And then we'll have another round of clinical signs and symptoms. So this is a reactivation. Yeah. And we'll see that this is a trouble, especially as we grow older, as our immune system starts to fade. It's also an issue if we undergo any kind of immunosuppressive therapy, say cancer therapy, transplant therapy, if we're on chronic long-term steroids. Right. So we'll come back to this case when we talk about the herpes viruses. And yeah, right. Okay. And another kind of connection here is that some of the latent viruses that cause the lifelong infections, some of them may also increase our risk for cancer, right? And actually, guys, we can get started on the second PowerPoint here. There are some additional slides on this PowerPoint that talk about mechanisms. So just the two key terms here, guys, latency, and then we're going to talk about oncogenic viruses. So the oncogenic viruses, right, they increase the risk of cancer. And the tough thing is that sometimes the latent viruses may also be, some latent viruses may also be what? Oncogenic, right? Yeah. So I just wanted to introduce Oncogenic, yeah, may also be oncogenic. So I just wanted to introduce those two terms. And folks, again, there are additional slides that go into some of the models for how viruses can cause cancer, and I'm going to skip those. Well, please forgive me, because what I want to do is like follow up on this whole concept of latent viruses and oncogenic viruses by switching to the second PowerPoint, where we'll actually have some specific examples. Okay, so I'm going to close down PowerPoint 1.